Okay, we're now going to hear from Martin Harvey about polling the pollinators taking part in the UK monitoring scheme. I'll let Martin introduce himself and his project and hand over to you now, Martin.
Brilliant. Many thanks, Kieran, and great to see everybody online. Thanks very much for inviting us to take part in the series this afternoon. So, as Kieran says, I'm going to be telling you about the UK Pollinator Monitoring Scheme, which we tend to refer to as PoMS, and about how you can get involved with that. There are a couple of main surveys that we run directly from PoMS and lots of other links out to other organisations that you can get involved with as well. See if I can persuade my slides to work. Here we go. I think everybody is sort of familiar with the idea that there is concern about pollination and pollinators and the potential declines of many of the insect groups that are associated with pollination. On a worldwide basis, of course, it's not just insects that carry out pollination. Lots of other types of wildlife play a role in pollination in different parts of the world. But in the UK, it is predominantly insects that do play this role. And we've all heard stories from all sorts of different quarters about the problems with insects declining and what that might mean for our ecosystems and for pollination in particular. And there certainly is evidence that we should be worrying about this. And back in 2016, the IPBES report looked at this globally and did highlight some of these problems, and also highlighted that obviously to address these problems, we need to be doing some conservation work, but alongside that, there's also a great need for monitoring of pollinators and their effects on the plants, partly to see whether our conservation work is succeeding and partly to try and focus effort in the places where it's really needed. So the concern, the need for conservation and the need for monitoring alongside that conservation has been well recognised for quite a while now, and does seem to have a considerable interest and support amongst members of the public. It's a topic that really has taken off, particularly around bees and pollination, and that's one of the sort of issues, if you like, that bees are just one part of the pollination story. Lots of other insects get involved as well, and the PoMS monitoring scheme is looking at all the insect pollinators. So what is PoMS? The Pollinator Monitoring Scheme grew out of DEFRA's National Pollination Strategy, which was launched back in 2014, which also highlighted the different strands of the conservation that was needed and the monitoring that needed to go alongside it. And PoMS was set up to address that monitoring need. And some of the things that are sort of different and special about PoMS is that it is taking a systematic approach to gather data on the abundance, so not just the occurrence and the distribution, but the actual abundance in terms of numbers of individuals of bees and hoverflies, and also the other flower visiting insects right across the board at a national scale. And it's the abundance data which has been really is quite hard to capture for insects generally. The number of insects you see is dependent on all sorts of things, including the weather, including the amount of time you spent looking for them and the method that you use to look for them. And it's one of the things that has historically been quite hard to assess is changes in the sheer abundance of insects. But we're not just doing something new and ignoring everything else that's happening. We are working very closely with the existing recording schemes for pollinating insects, and we see the PoMS, the data that PoMS is collecting as new is very much an addition to the long-standing and excellent history that we have of recording things like bees and wasps and hoverflies through the recording schemes. And by adding that long-established distribution and occurrence data to these new systematic and abundance surveys, we hope and we think that we can actually produce some more informative information on what is actually happening and what is actually changing in the populations of pollinators. So PoMS is directly linked to the pollinator strategies in the UK countries, and in 2021 Northern Ireland has joined the partnership as well. And across the Republic of Ireland there's the fantastic All-Ireland Pollinator Plan happening at the same time. So lots of activity, but also lots of links being made between these things. So moving straight on to the most direct way that you can take part in PoMS is through what we call the FIT counts, and that stands for the Flower Insect Timed Counts. And I'm going to tell you a little bit about that and how you can get involved, and then we'll have a go at identifying some of the insects for these counts. So basically what the FIT counts ask you to do is to find a patch of flowers. You do need to wait for good weather to do it. So there are temperature thresholds. The weather for the last few weeks has been sort of hovering around the thresholds a little bit. So it's been a little bit difficult to actually find the right conditions. But once you have got good weather, you find a patch of a particular type of flower. You need to define a 50 by 50 centimetre patch, count the number of flowers in the patch, and then spend 10 minutes counting all the insects that actually land on the flowers. So not the ones that land on the leaves, not the ones that fly in and have a look and fly away again without landing. The rules for the FIT count are to count all the insects that actually land on the flower, even if they only do so briefly. And having done that for 10 minutes, the results then need to come into PoMS, and that has been via, well, in fact, up until now, they come in via a form on the iRecord website, but we will very shortly, hopefully within the next few weeks, have a FIT count app that you'll be able to use, and also a new website interface that is on the way as well. I'll say a bit more about that later on. So in terms of preparing for the count, we hope that it is a reasonably simple exercise to do and an enjoyable one, and the feedback that we've had so far suggests that people do find it enjoyable to do. But there's a little bit of preparation you need to do. You need to find a way of marking out your 50 by 50 centimetre square. And it's possible to purchase quadrats that give you a 50 centimetre square, but it's also very easy to make one yourself. And people do that in all sorts of ways by using lengths of cane or lengths of string, or we're quite fond of these gaffer tape homemade quadrats, where you measure out a piece of gaffer tape and fold it over to make a good sort of strong and slim edge to your 50 by 50 centimetre square. The advantage of the gaffer tape ones is that they can be scrunched up and put in your pocket and carried around with you. So we use those quite a lot. Doesn't matter how you do it, as long as you're defining a square of the right size. Then you need to find some flowers to use for your FIT count. And we have this list of 14 target flowers, and they're listed more or less in order of the time of year that they appear. So there's obviously a sequence through the year of different flowers that you can use. And we always like to encourage people to do FIT counts on one of the target flowers if you possibly can. And the reason for that is simply so that we get a good lot of data on this particular set of flowers, which just makes the data more comparable, and we can do the analysis in a more consistent way if we get lots of counts from these target flowers. However, you may be in a place where none of the target flowers grow, or it may just not be the right time of year for them to be growing near you. And in that case, it's absolutely fine to use other flowers. So if you want to do a FIT count in your garden and you haven't got any of the target flowers, but you have got other things flowering, then just choose another flower. Tell us what it is, and you need to take a photo of the flower, but that's absolutely fine if you can't do one of the target flowers. There is a recording form which you can download from the PoMS website, and if you've got access to a printer, which of course not all of us have at the moment, then you can print that form out and use it in the field to actually capture your counts. But once you get the hang of what's needed, it's fairly easy just to write it in a notebook, and of course once the app comes out in a few weeks' time, then you'll be able to use that as an alternative as well. So as long as you're capturing the set of information that the recording form needs, again, it's up to you how you choose to do that. So as I say, we need to have decent weather for pollinators, and we've defined that as if the sun is out and it's at least 13 degrees, you're good to go. If it's cloudy, then we need it above 15 degrees to carry out a fit count. And those thresholds are designed just to make sure that there is insect activity going on at the time you do the count. So as long as those temperature thresholds are met, you can do a count at any time from the beginning of April to the end of September, and that will all get included in our analysis. And you can just do it as a one-off. You could just decide to go out and give it a try and send that in and then stop, and that would be a good contribution. But of course, if you can do more than one count, and particularly if you can do a number of counts through the year at different times, there's no set pattern that you have to follow. You don't have to do it once a week or once a month or anything like that. It's up to you. Some people have chosen to do it very regularly, once a week or even once a day or even several times a day in some cases. But as I say, there's no sort of rule that you have to follow there, whatever works for you, and all contributions are very welcome. The thing about fit counts is that they're one of these surveys that the more people that get involved, the more counts we have coming in, the stronger the data will be. So it is really something that we're encouraging as many people as possible to have a go at. This is just a particular count that was summarised for us by one of our wonderful volunteers, Edwina, who's been volunteering for a number of years with us now. And she put together this slide from one of the fit counts that she'd done, just to sort of pick up on some of the things that she found either of interest or that she had to stop and think about. So obviously you need to record the date and time that you're doing the count. You have to count the number of the target flowers that are within your 50 centimetre patch. And counting flowers sounds like quite an easy thing to do, but it actually raises quite a lot of questions about exactly what it is that you're counting, and indeed exactly what it is that is a flower. And looking at this particular plant, the cow parsley, you could start to try and count all the tiny little white flowers individually, but you'd probably be there quite a long time doing that. So for cow parsley and the related umbellifer type plants, it's actually those umbels, the sort of groups of white flowers, that we ask people to count. And each target flower has got its own type of count, so that for dandelions, for instance, you're actually counting the single yellow flower head, which technically consists of lots of individual flowers, but it's one flower head, and it's the flower heads we ask you to count. Whereas for something like lavender, we ask you to count the flower spikes rather than the individual flowers. So there's a little bit of thought that needs to go into how you actually do the flower counts. But as I say, we do also ask you to take a photo of the flowers, which allows us to do a check and to make any corrections needed if they are necessary. And we ask you to say whether your flower patch is growing in a big patch of the same flower, or in a patch with lots of other flowers, lots of different flowers, or whether it's growing more or less isolated. So there's also some questions about the general habitat in the area that you're counting as well. So then getting on to the count itself, as I say, you set your timer going, and during the 10 minutes, we do ask you to try and count every insect that lands on the flower. And what we ask you to do is to identify them into main insect groups. So we're not looking for species records here, we're just looking for groups, and we're going to do some practice on the different insect groups in a moment. So a lot of insects you'll probably be able to put into their groups quite quickly and easily, but there are some trickier ones as well, as we shall see. We ask you to count each individual insect once, and to try not to count the same individual several times over, but that is one of the things that's actually much easier said than done. And if you go out on a busy summer's day when there's lots of insects flying backwards and forwards, that is quite a challenge, is how you try and confine yourself to counting each individual once. And we recognise that it's going to be impossible to make that an absolutely perfect count, but all we can ask you to do is to make your best judgment that you count each individual insect once. So if you see that an insect has flown in and you count it and then it flies away again, and then another one flies in from a different direction or something, then that's probably going to be a different insect and it should be counted as number 2 on your list. But if you see that it just takes off from a flower and then lands again immediately, then that's the same individual and that shouldn't be counted again. So as I say, impossible to make that perfect, but give it your best judgment. And the other point that Edwina's highlighted is that we do not ask you to take photos of the insects during the count, mainly because we know that if you pick up your camera and go towards the flower or lean over the flower, that does disturb the insects and affects the pattern of the ones that are visiting the flower. So we do specifically ask you not to do that during the 10 minutes. However, at the end of the 10 minutes, you can stop and take some photos of the insects and add them to your count if you wish to, or if you want to actually do some species recording, you may need the photos to do that. And that's absolutely great and fine, but don't do it during the 10 minutes, please. So the PoMS scheme started up in the middle of 2017, and as you can see from just immediately looking at these charts, the numbers of people involved has been growing since then. We are, as we speak, my colleagues are in the process of analysing the first 4 years worth of data that PoMS has collected. And I'm not yet in a position to share a lot of those results with you. They are still being analysed and presented, but they will, this initial 4 years worth of analysis will be coming out in the not too distant future, and we'll make sure that's shared with everybody involved with PoMS when that comes. But this just gives you an idea of the overall participation, and as you can see, the numbers have been growing. We had nearly 2,000 counts in 2020, which was fantastic. There did seem to be an interesting effect of the lockdown situation combined with the excellent spring weather that we had in 2020. And if you remember, last April was much, much warmer than it has been this year. And that certainly got the fit counts off to a really good start last year. Numbers have been much lower for April this year because of the weather, but we're hoping that things will pick up. And yeah, as I say, we really want as many people as possible to participate and submit their counts. That would be fantastic to see the numbers continuing to grow. One bit of analysis that we have been circulating for a little while now are these what we call donut charts for the different target flowers, which give you an idea of how many insects you're likely to see, and also which flowers seem to be most associated with the different insect groups. And these might be a bit difficult to read on the screen, but what we're looking at on the left here is the chart for dandelions, which is obviously a flower that is out now. And I've been doing quite a lot of counts on dandelions the last couple of weeks. And on average, if you do a 10-minute count on dandelion, you can expect to see 4 insects during your 10 minutes. And quite a lot of those insects will be in that sort of grey, sorry, I just went, go back to the slide. Yeah, that sort of large grey band there are the small insects. We have a category for just the really tiny insects, things like pollen beetles and tiny flies and tiny wasps that are really not possible to identify accurately without peering at them very closely. And dandelions do attract an awful lot of those. I've seen a lot of pollen beetles on my dandelions recently. But there's quite a spread of other things as well. Flies are quite attracted to dandelions, and you do see some of the bees coming in as well. On the right hand side of this screen, we've got the equivalent chart for white dead nettle, which is also in flower now, and which also attracts an average of 4 insects per 10-minute count. But the makeup of the insect groups is quite different for white dead nettle, and it's bumblebees are particularly fond of white dead nettle. Those quite deep flowers are sort of designed, or evolved at any rate, for bumblebees to dig their way into and to reach the nectar that's right at the base of the flower. So they tend to be a good feature of the white dead nettle. But again, you do get a range of the other insect groups visiting from time to time. And just a couple more examples of these charts. So buttercup on the left, another one with quite a lot of tiny insects, quite a lot of flies, and a mixture of the other groups. And hawthorn should be in flower in the not too distant future, which gets 6 insects per count on average. Lots of flies, quite a lot of hoverflies, quite a lot of honeybees get seen on hawthorn. They stand out quite well in the chart there, the sort of orangey colour to the top right of the circle. And as we get further with the analysis, we hope to make a lot more of this sort of information available so that you can get an idea of how your counts are comparing to the counts that are being done elsewhere, or the particular time of year, the particular flower, the particular part of the country you're in. There's all sorts of variation that could crop up in the counts. And as time goes on, we're going to try and make more and more of that information available to you so you can see how your counts are comparing. Okay, so that's what the fit count consists of. And when we get to the end of the presentation, if you've got any questions on that, please do make a note of them and pick them up at the end of the presentation. And please do spread the word. As I say, we are keen for as many people as possible to have a go at doing this, and hopefully to enjoy doing it, and carry out repeat counts as and when you can. So we're going to have a look now at the different insect groups that we ask you to group things into. And some of these, as I say, are quite obvious. For instance, butterflies and moths. We only ask you to recognize them as butterflies and moths. You don't even need to know whether they are a butterfly or a moth. They're all treated as one group for the Lepidoptera. And I think most people would quite happily recognize a butterfly as being a butterfly and be able to put it straight into the right category. But there are one or two others that are a little bit more tricky. And we do have some identification materials on the PoMS website, and that some of the slides that I'm about to show you are included in the insect ID guide that we have up there. PoMS is not directly aimed at monitoring honeybees. We are really more focused on the wild bees. But of course, people do see honeybees landing on flowers, and we are also keen that people start to realize the distinction between the different types of bee. And we all know that there are sometimes confusions in newspaper stories and so on about what's happening to honeybees as opposed to what's happening to bumblebees or solitary bees. So part of the sort of purpose, if you like, of PoMS is to get people a bit more familiar with these different categories. And honeybees are just one species in Britain, but it is quite a variable species, at least in terms of the colour on the body, and it can be quite tricky sometimes to pick out honeybees from the solitary bees in particular. So we've tried to highlight some of the features here. One of the best features, certainly for the worker honeybee, is that hind leg, which has that very characteristic flattened, broad shape to it. And that's where, when the worker honeybees are collecting their pollen, that's where the pollen gets stored in that pollen basket on the hind leg. And that's definitely something to look out for with the honeybees. The other thing with all bees to look out for is the antennae, which are relatively long in all bees. And that's a really good distinction from some of the hoverflies, as we shall see later on. There's a few more slightly detailed things you can look out for, such as on the wing margin there, you can see that long and narrow marginal cell. That's the sort of long oval shape that the wing veins outline just by the edge of the wing, and that's quite a good characteristic for honeybee as well. So that's some of the information that's in the ID guide that you can download. And then we have 20-something bumblebees and 200 and something solitary bee species in the UK to look out for. And again, as far as the fit counts are concerned, you don't need to know the species, but you do need to be able to sort them into one category or the other. And a lot of the time, bumblebees are reasonably, most people, I think, are comfortable with recognizing a bumblebee. They have that very rounded, very hairy sort of appearance. And most solitary bees are a bit smaller and a bit more elongated and a bit less hairy. But there are one or two confusion species. There are one or two solitary bees that are quite rounded and can end up looking like small bumblebees. And indeed, the smallest bumblebee workers are smaller than the largest of the solitary bees. So you do have to sort of look quite closely at some of these things. And for me, that's one of the really nice things about doing fit counts, is that you do get to spend 10 minutes peering really closely at insects and getting familiar with them. And it's not just the way they look, it's the way they fly, the way they behave, even the way they sound. The different sort of buzzes that you get from the different species are something that you can start to pick up on. And having that 10 minutes of focus on doing that, to me, is a really sort of relaxing and intriguing, interesting thing to do. So we're going to have a go at trying some of these out, and I'm guessing that this first one might be quite straightforward, but if we just take a moment to look at it and check the sorts of things we've been talking about to do with the body shape and the length of the antennae and how hairy things are. So just spend a moment thinking about that, and then I'll ask Kieran to put up the list of options as to what you think that insect was that we were just looking at. I believe you can move this poll screen out of the way if you want to be able to see the photo while the poll is up there. You can shift it around on your screen, but yep, doesn't look like it's a lot.
You also won't be able to see all of the options in one go, so you might need to scroll down to have a look if the option that you want isn't at the top.
Good point, Ken. So there's one more option that I think is off the bottom of the list, and that's for the other insects. And I'll come back to why you might want to use other insects in a moment. Okay, but we've got quite a lot of these to have a go at, so we need to be sort of fairly quick on the draw, but I think we're ready to go. And okay, we have one vote for solitary bee and a lot of votes for bumblebees, and this one is indeed a bumblebee. So if you can take the results off the screen, Kieran. Do you do that, or do I do that?
I've stopped sharing them now, so I don't know if they've disappeared for people, if they have to.
They stayed on for me, but I can switch them off anyway, so that's fine. So yeah, so this one was indeed a bumblebee, and yeah, it looks like a bumblebee if you know what bumblebees look like. It does have those long antennae. It is very hairy over the body, and it has that quite rounded shape. And as I say, there are some solitary bees that do have a similar shape. Most solitary bees are a bit longer and thinner, but not quite all of them. And rather few solitary bees are really as hairy as this one. But again, there are one or two exceptions. So it does take a little bit of time to a little bit of experience, I guess, to really pick these out. But if you spend some time watching bumblebees and getting used to the way they fly and the size of them and just the way they behave around the flowers, I think most people will be fairly comfortable with picking out the bumblebees. Okay, let's try our next example. So back to the poll again, please. Well, after a second, let's give people a second to stop and look at this one.
Okay, you should be able to vote again now. Just to let everybody know, the polls are anonymous, so we can't see who's voting for what. So even if you're not completely sure, it's worth having a guess. I can let you all know that this is not as unanimous as the last one. It's a lot more divisive, this one. But yeah, even if you're not sure, have a vote, see what you get. All right, we'll give it 4 more seconds. Yeah, okay. There we go.
Okay, so that was a much closer match, wasn't it? And the correct choice has won out. It is a solitary bee, but that was obviously a much harder one for people to pick up on. And I'm not terribly surprised by that. It was a rather trickier one to deal with. So why is this a solitary bee and not a honeybee? Let's have a look at that. So it has the long antennae, but so do honeybees. It has a relatively narrow body shape, which is different from the bumblebees, but quite similar to the honeybees. It is actually quite a small bee, this one, but that's very hard to judge from a photo, of course. You get a bit of an idea from the plant that it's sat on, but it is, I can tell you it's smaller than a honeybee, but you can't really see that. And so what does pick it out from the honeybee? It is a little bit hairier, particularly on the abdomen, than a honeybee would be. But the really definitive thing is that hind leg, and it's not as broad and flattened as a honeybee, but it is covered with lots of hairs that a honeybee wouldn't have. And the thing that can make this confusing is because the hairs that we're looking at on this picture have a few specks of pollen. I don't know whether you can see that, but there are some little yellow pollen grains stuck to those hairs, and that is the pollen part of the solitary bee's pollen basket, and she does collect up pollen there. But the hairs themselves are not present on the honeybee. The honeybee has a more smooth, shiny surface on the hind leg, and we'll come back and have a look at another honeybee example in a little while. So there is a difference in that hind leg, but because it's covered with hairs, it's more difficult to notice whether it's as broad and wide as the honeybee or not. But in this case, it isn't, and that's really probably the most straightforward thing to pick up on as being the difference. But that is quite a small difference. I'm absolutely accepting of that. It's not the easiest thing in the world to do sometimes. And in terms of the results of the fit count, we do realise that people will make mistakes, and some of the distinctions are much harder to do than others. And we have to accept that not every result that comes in through the fit counts will be perfect. If you are able to add some photos after your count has finished, you can add some examples of the different insect groups that you've counted as photos with your count. And we do some checking of those to get an idea of how often people are putting things in the wrong category. But in the end, what we're really interested in out of all this are the numbers, and we accept that there will be some variation in the groupings, but the total numbers that we get through, particularly the more people we get involved, the more those numbers should still be adding up to tell us something about the total numbers of insects that people are seeing. So let's move on to the next one, and just a couple of moments looking at that one, and then we'll see whether the voting is any more decisive on this one.
Okay, the poll's going to come up now, guys. Again, it's anonymous, so even if you're not sure, have a vote. Okay, we'll give you 10 more seconds. Three more seconds, 2, 1. Okay, we've got quite a lot of people. There we go.
Okay, so, well, this one is a honeybee, and honeybees have won by some considerable distance in the poll, but we've got the opposite of what we had last time with the solitary bee coming back into this, and also a few other categories being suggested as well. So the balance here has been for honeybees, which is absolutely correct. So let's have a look at the image here and see if we can pick out some of the things that make this one a honeybee. So, well, first of all, starting with the fact that it's a bee as opposed to anything else, the antennae are, again, a really good clue for that. So there aren't any hoverflies that have antennae as long as that, and with so many segments. That final bit of the antennae, the sort of finger bit of the antennae, is made up of 12 or 13 segments in a bee, and there aren't any hoverflies that have such long segmented antennae. So that sort of rules out, that's one of the things that rules out the hoverfly straight away. Difference between bees and wasps is actually a bit of a trickier one, which we're going to come back to in a moment. But it's quite a hairy insect, this one, and most wasps are not as hairy as this. So that brings us back to what type of bee is it? And it's not really the right shape for a bumblebee, not really hairy enough for a bumblebee. It could be seen as a solitary bee, but this time we do have a view of that lovely broad, flattened, shiny hind leg. And we can also see that really long, narrow sort of tube in the wing veins at the edge of the wing. And both of those things suggest that we are looking at a honeybee in this case. Quite a dark honeybee. Some honeybees have much more orange on the abdomen than this one does. So this is quite a dark example. But there it is a honeybee. And I think I've got a couple of things. So we saw this photo briefly earlier on. This is a honeybee that started collecting pollen onto her hind leg. And one of the things that distinguishes the way honeybees collect pollen from the way that solitary bees collect pollen is that honeybees do so by moistening the pollen and collecting it up into quite a solid and actually quite shiny ball on the back leg. So if you look really closely, and you might have to sort of catch an insect in a pot and get it under a hand lens to see this, but if you look really closely at that hind leg, the honeybee's pollen basket contains this very solid and shiny pollen thing. Excuse my phone, I should have turned that off before I started, and I can't work out how to stop it. So whereas with the solitary bees, they do collect pollen on the hind legs in a similar way sometimes, but they don't moisten it and they don't collect it into such a solid ball. They have it as a sort of much looser lump of pollen. And although, as I say, you need to peer quite closely to see the details of that, I think it is something that you can pick up on after a little while as to whether you're looking at a honeybee with its solid lump of pollen. Bumblebees also have a quite solid lump of pollen, whereas the solitary bees have a much looser sort of dusting of pollen, although that dusting can build up to be quite substantial. And I think there's one other example of the honeybee next to one of the hoverflies. So here we're looking at the honeybee on the left and the hoverfly on the right. And the hoverfly is the type of hoverfly that gets called a drone fly. And it's called a drone fly because it does look very similar to the honeybees, which of course have drones as the males of the honeybee. And at first glance, they really do look like very similar insects. But if you can see the antennae there, we've got, again, that lovely long sort of finger-like antennae for the honeybee and much shorter, stumpier antennae from the hoverfly. The hind leg, broad and flattened for the honeybee. It's a little bit broadened in this particular hoverfly, but it's not that same sort of flattened shape that comes out to a sort of flat edge at the end. There's some difference in the wing venation, if you get a chance to see that. And the other thing is the eyes. I mean, honeybees have fairly large eyes, but hoverflies have really big eyes. And they can cover most of the head. It varies between the males and the females. Males have slightly bigger eyes than the females in the hoverflies. But even in the female hoverflies, their eyes do cover a substantially larger part of the head than the bees' eyes do. And that's something that I know a lot of people look out for as well, to try and tell those two groups apart. But for me, the most obvious thing is the antennae to look at first, just to get those two sorted out, those two groups sorted out. And just to pick up on the insect ID guide again, to emphasize some of those points, I think so hoverflies as a group are quite varied. There's quite a lot of the black and yellow hoverflies that are seen as wasp mimics, but there's also some of them that are very good bee mimics as well. And we're looking at the dronefly example again there that we saw just now. And always check the antennae. There are some other differences, and one of the things that all the books will tell you is that flies have one pair of wings, whilst bees and wasps have two pairs of wings, and that's absolutely true, but it's quite hard to see on a living insect in the field. The bees and wasps often have their wings overlapping or even rolled up a little bit, and it's not always very easy to tell whether you're looking at one pair of wings or two pairs of wings, but that, if you get a good view of the wings, that can certainly help as well. But the antennae and the eyes, I think, give you the first clues. The behavior as well. Hoverflies are called hoverflies for a reason, and they often do hover around flowers. And with one or perhaps two exceptions, bees don't hover in the same way. So the behavior can give you a good clue as well. And if you're looking at a female bee and she's collecting pollen, that's another good clue that you're looking at a bee, because hoverflies don't collect up pollen onto their legs. And wasps don't collect up pollen in the same way either. So if you're seeing a clear pollen basket, then again, that's a good indication that you're looking at a bee. And in the hoverfly wings, which I accept you don't always get a good chance to observe, but if you do get a good view of the wings, there are some good characters there. the distinguishing feature of, or one of them, of the hoverfly family is something known as the spurious vein, the vena spuria, which again is actually quite hard to see in the field. But there's another one that's a bit easier to see that isn't exclusive to hoverflies, but is a good clue for them, is where you get this so-called false margin just in from the wing edge. It's sort of parallel to the edge of the wing. There's a clear line of veins there, which a lot of hoverflies show quite nicely. So that's something that bees don't have in the same way, and that can be quite a good clue as well. Because we're talking about whole groups of insects, and there's lots of different species within the groups, there's always exceptions to all these rules that I'm giving you. So you will every so often encounter something that doesn't follow the rules, but for the most part they will do. And once you get your eye in for their behavior and their appearance, it does become easier over time as you build up your experience. So we've seen the dronefly again, and the bottom pair of insects here. We have a hoverfly on the left, bumblebee on the right. There are some hoverflies which are absolutely fantastic impersonators of bumblebees, but once again the antennae do look quite different. So if you can get a good view of the antennae, that's really important for telling those apart. And if you want to pick out droneflies as a group within the hoverflies, they have another little wing vein character, which if we zoom in a little bit, you can see that little sort of loop in the wing vein here is very characteristic of the particular set of hoverflies that the dronefly belongs to. And if you can see that in the wing veins, you're definitely looking at a hoverfly and not at a honeybee. Okay. And then there are lots of other flies that aren't hoverflies, and you will see a lot of other flies on flowers. There's getting on for 7,000 species of flies that aren't hoverflies. And for PoMS, we don't need to know which of the 7,000 it is. We don't need to know what type of family it belongs to. We just need to decide whether we're looking at a hoverfly or an other fly. And the other flies are enormously varied in their appearance because there's so many of them there. But of the ones that you see sitting on flowers, you see quite a lot of these, this group known as calypterate flies that are generally very bristly, much more bristly than hoverflies generally are. And then you get all sorts of other shapes. And your task is to ask yourself whether you're looking at a hoverfly. Does it behave like a hoverfly? Does it have that typical hoverfly look to it that you'll get used to after a while? Or does it look like something different, in which case it goes into the other flies category? Okay, a little bit of practice on these then. So take a few moments to look at this insect. Think about antennae and wing veins and all the other things we've been talking about. And then we can open the polls for this one.
Right, so we've got 20 more seconds. Remember to look at the antenna, the eyes, the wings. Yeah. 5 more seconds. Almost got everybody. Come on. Just a few of you left.
Okay. So a clear vote for the hoverflies there, which is absolutely right. Yeah, this is a hoverfly. It is, again, the dronefly that we've been talking about. And really those antennae should exclude it from being a bee or a wasp. And if you could see its behavior, that would help you judge that it was a hoverfly, not an other fly. And that, if you can see the little loop in the wing veins, that's another confirmation that it's this particular type of hoverfly. So yeah, not too much disagreement about that one. How about this one? Take a few moments to have a quick look at the overall appearance. Okay, quick vote on this one. Perhaps we aim for about 20 seconds on this one. You know, people are getting the idea of the voting. We get most people in, I think. And Yep, and again, the majority vote is definitely right on this one, that we are looking at an other fly. And so for PoMS, you don't need to know that it's a fly from family Tachinidae, but that's what it actually is in this case. But for PoMS, it's just an other fly. And this is a fly that does actually have slightly longer antennae than most flies do, but the antennae are still not as long as a bee, and they certainly don't have all those individual segments that a bee's antennae has. So there is scope for a little bit of confusion over the antennae here, because they are longer than the average fly, but they're still not as long as a bee. And within the flies, this is clearly a very bristly fly, which just about excludes it from being a hoverfly and puts it into one of the other categories. And this particular tachinid fly is one that you see very commonly on flowers in late summer. So where are we going next? Let's not do a vote on this one, actually. Just think to yourselves what you think this might be. And again, from photos, it's always hard to judge the size, but this is one of the quite tiny insects that you might see on a flower. You might get an impression of that just from the fact that it's sitting on one petal. I can't actually remember what this flower was, but it's clearly, the fly is much smaller than the flower, and it really is a tiny species. And I don't know whether you guessed to yourself correctly, but this is another type of hoverfly. And it does, you can just about see through the wings that it does have the typical black and yellow wasp type markings that hoverflies often have. It's not a wasp though, because its antennae are too short. And it's the difficulty with the wing venation here is that the two wings are overlapping each other, but you can just about make out that it's got that false wing vein along the edge of the wing there. But the head has got a very typical hoverfly appearance to it, with those enormous eyes. This one is another female. She has a gap between the eyes, whereas if it was a male hoverfly, the eyes would meet up in the middle. But it's that quite sort of shiny head with enormous eyes and short antennae is definitely a good hoverfly characteristic. Okay, we better move on a bit, otherwise we're going to run out of time today. Bees and wasps are, again, if you're looking at a bumblebee on the one hand and a typical social picnic wasp on the other hand, they are obviously different shapes and sizes and different appearance, and most people tell them apart. But technically, the difference between bees and wasps is quite a hard one to really define. And one of the things it comes down to are the hairs on the body. Bees tend to be hairier than wasps, but wasps do have hairs. And if you get them under a high-powered microscope, the difference is that the hairs on a bee have little branches on them, and the hairs on a wasp are just single hairs. But of course, that's absolutely hopeless for recognizing them in the field. You just can't see that unless they're under a microscope. So we have to go try and make a judgment on the general appearance and shape. But it is true that on the whole, most bees are more hairy than most wasps. Some of the wasps, and again, there are a lot of different species of wasp in Britain, but there are a few that are these big black and yellow ones that we think of as picnic wasps, but a lot of solitary wasps as well. And some of them actually roll their wings up. You can see that in the wasp on the right here, that the wings are actually folded up when it's completely at rest. But when they're not at rest, the wings will be held open and look much more like the bee's wings. So that isn't always a helpful thing. And I have to admit that the bee and wasp distinction is the one that still catches me out quite a lot, particularly when you think that there are some quite small solitary bees and some quite small solitary wasps and some really dark blackish species in both categories. And this is one that we know people will struggle with from time to time. You're much more likely to see a bee on a flower than a solitary wasp on a flower, but solitary wasps do visit flowers and you will see them from time to time. Bees, even just looking at these two tiny examples here, the bee, I think, is obviously or distinctly hairier than the wasp, so you've still got that to pick up on. Bees often have some sort of marking on the abdomen, but not absolutely always. You do sometimes get some completely black bees. But if it's a female bee, she'll definitely have hairs somewhere on the legs or body in which she'll be collecting pollen, and I say wasps do not collect pollen. So that's a good thing to look out for. And the other thing with a lot of solitary wasps is that their heads are that quite rectangular, boxy shape for quite a lot of the species. And I think that's quite a good clue to pick up on as well. Another type of wasp that, as far as the fit counts are concerned, also goes into the category wasp are these long, thin creatures that are known as ichneumon wasps. And they're a different part of the Hymenoptera. They are parasitic creatures that lay their eggs into other insects. And most of them are this very long, narrow shape, and nearly all of them have very, very long antennae made up of an enormous number of tiny segments. So even more than the bees and wasps, the bees and the typical wasps have, these ichneumons have these multi-segmented long antennae. And if you see a female ichneumon, they often have a long ovipositor as well, but obviously that's only in the females. You don't see lots of them on flowers, but some of them are very regular flower visitors, and you'll probably encounter them sooner or later. So the long, thin body and the very long antennae should help you pick these out, and they go straight into the wasp category alongside the other wasps. And the final type of Hymenoptera that causes confusion, which you will see on flowers occasionally, are the sawflies, which are a bit more distantly related to the bees and wasps, but they can look very wasp-like. They're generally not hairy creatures. They do have quite long antennae that look quite similar to the typical bees and wasps. And the sort of real distinction for sawflies is that they don't have that pinched wasp waist between the abdomen and the thorax. They're more or less a sort of constant tube from the abdomen up to the thorax, and there's no narrow bit in the middle. And if you can get a good view of that, that's the thing to really look out for sawflies. But again, this is one that we know people do struggle with from time to time, and sometimes you just can't be certain, which means you either have to make your best guess and put it in the category you think it is, or if all else fails and you're just not sure, it can go into the other insects category, and sawflies go into the other insects category anyway. But anything that you're just not sure about and can't make a decision on can also be counted as an other insect as far as the fit counts are concerned. What we don't want you to do is to completely ignore an insect. So if you see an insect and you just don't know what it is, please still count it into the other insects category. Okay, so we've got our final few examples to have a go at, and I've included one or two slightly tricky ones in here from all the different groups we've been looking at. Let's do a vote on this one if we can, Kieran. Just a couple of seconds more to look at it. Oops, hopefully people saw enough of that. Plenty of votes coming in. Okay, I think we're nearly getting that. Okay. Okay, a clear win for solitary bees in this case, with a few votes for a number of other categories. And let's see if we can. So this is a solitary bee. It's a slightly different group of solitary bees that we haven't looked at so far today, but you can just about see the long antennae, which exclude the hoverflies. It is quite hairy on the head and the top of the thorax. The abdomen's not so hairy on the top, but it has got a lot of hairs underneath, and that degree of hairiness is pointing us towards bees. Doesn't really look the right shape for a bumblebee. It's too hairy for a honeybee, or at least the hairs are in the wrong places, and it's fairly elongated, and that all puts it in the solitary bee category. And this is actually one of the leafcutter bees, and the group of solitary bees that include the leafcutters and the mason bees don't have a pollen basket on the legs. They actually use these lovely hairs underneath the abdomen is where they collect up their pollen. And if you see a bee-like insect that has got lots of hairs and pollen on the underside of its abdomen, then you can be sure you're looking at a solitary bee and that you're looking at a mason or leafcutter bee within the solitary bee grouping. So that's a nice little one to look out for. Okay, we won't do a vote on this one, but just have a quick think to yourself about what this might be. hopefully most of you will rapidly come to the conclusion that it is a fly rather than a bee, and it is in fact a hoverfly rather than another sort of fly. And again, short antennae, enormous eyes. If you get a good close view of a hoverfly, a lot of them have this sort of distinctive head shape with a nose and with what looks like sort of pouted lips. They aren't really lips, of course, they're just the shape of the front of the head of the hoverfly. But that sort of bumpy shape is quite characteristic of a lot of the hoverflies if you get a good clear view of it. And less hairy than the bee, a bit more shiny than most bees, perhaps just about make out the wing vein, the false edge to the wing there. So we're definitely looking at a hoverfly in this case. Right, I'm going to give you a vote on this one. What do you? There's a few seconds to look at it. Think about shape, think about hairiness, and then let's give that a try, voting wise. Well, see, people seem a bit reluctant to commit to this one.
You're going to have to, because they're going to make this a quick one, guys. You've got 5 seconds.
Yeah, we are running out of time, aren't we? I will speed up through the next few.
Yeah.
Okay, I think we're just about there for that, are we? Brilliant. Okay, well, I'm impressed. That's a really clear vote for wasps, and you're absolutely right with that. So that's good stuff. It's got black and yellow markings like a hoverfly would have, but the antennae are too long for a hoverfly. It's not very hairy, or at least not obviously hairy, and it does have that boxy head shape. And yeah, you're absolutely right. It does also have a bit of a wasp waist here as well, all of which adds up to it being one of the solitary wasps. So well done with that. Right, we are going to have to move on. This is quite, I think this is quite a tough one, actually. So just think to yourself, we won't do a poll on this one, but just think to yourself what this is. It's actually, oh, we are doing a poll. Have you stopped that again?
Yeah, yeah, there's no poll, there's no poll.
Let me just close it. Yeah. So this one, in terms of colour, it's got quite bleached, and this is one of the things that happens to bees as they get older. Their hair colours tend to sort of fade and get a bit bleached, and this one does look a bit odd. It is in fact a honeybee, and it to me doesn't obviously look like a honeybee just in general appearance, but we can see it's got that very broad hind leg and it's got that long cell at the wing edge, and it is in fact a rather dark-bodied and pale-haired form of a honeybee. So you really do need to key into those legs and wing venations if you can possibly see them. This one is an other fly. It's a group of flies known as dance flies, many of which have this spectacular long proboscis, quite bristly again, doesn't really look much like a bee, this one, I hope, and it's got quite short antennae. A really good advert for the role that flies play in pollination. I think here the fly is absolutely covered in pollen under its body, and the fly, of course, is not collecting the pollen. Bees are collecting pollen up and taking it away for their own purposes, whereas flies are just collecting pollen and then going on to visit the next flower. So they do actually do a pretty good job of pollination, and they are a very important part of the whole process. This is the one that we spoke about a little while ago as being one of the tricky ones to pick out, but it's that body shape, no wasp waist. It's like a tube continually up the abdomen and into the thorax without any constriction, which means we're looking at a sawfly in this case. And we mentioned that there is an other insects category for anything that you can't identify or that doesn't fall within the other groups. And we also have a small insects category, which is you often see on a flower that it can sometimes be absolutely packed full of really tiny insects. And by small insects, what we say are things that are 3 millimeters or less. And typically that will include pollen beetles and some of the tiny parasitic wasps and some tiny flies, and you occasionally get aphids sitting in flowers as well, all of which you don't need to try and distinguish in any way other than to say that's a small insect and it goes in the small insect group. There are some small bees, but they're all bigger than 3 millimeters, and they should still be counted as bees. But anything really tiny, you don't need to try and distinguish. It just gets counted as a small insect. Right, I'm aware that I'm pretty much out of time, but I have got some more stuff I wanted to continue with. So as long as Kieran's happy for me to go on for a little bit longer, I will try and get into the next bit of the talk. But thank you for participating in that. Hopefully that has intrigued you as to how you tell insect groups apart, and hopefully it hasn't put you off too much, but it does, I think, make it clear that some things are really obvious and straightforward. Other things do take a bit of practice, and taking part in the fit counts and having a go at them and keeping your eyes open and looking at your insects and paying them attention is a really good way to start learning your insect groups, I think. And I hope you find it an interesting challenge rather than something that's too off-putting. Okay, then I will try and quite rapidly move through the last couple of sections that I wanted to cover. And the fit count is the sort of main public bit of the PoMS surveys, but we also have another much more systematic survey, which is also very much supported by our wonderful team of volunteers. So I'm just going to quickly tell you something about this. The 1 kilometre square surveys happen in just a particular set of 75 1 kilometre squares that are scattered across the country. And we're hoping that Northern Ireland is going to get up and running with this year as well. So they can only be done in these squares. We can't, I'm afraid, set up new squares, and there are large parts of the country where we don't have a square nearby. So I'm afraid whether you're able to join in with this survey does partly depend on where you live. But we do have some fantastic locations for them. Some of them are in these gorgeous upland sites. Some of them are in rather lovely bits of lowland woodland. And some of them are in bits that some people might think are a bit less lovely or interesting in agricultural habitats, but they're still very interesting in terms of what insects you actually find in them. So there's a real mix of different squares that we sample in. And what the survey actually consists of, this is very different to the fit counts. It's a survey where we ask people to put out traps, and these are traps that are actually collecting and killing insects. It is a lethal trapping method. So it's been designed to capture the minimum amount of material that we need to give us some robust statistical results as to changes in pollinators across the whole country. And part of the reason, of course, for actually trapping insects in this way is so that they can be brought back to the lab. And for the bees and hoverflies, they are identified right down to species across the whole country for every single specimen we collect. And the other specimens are counted into their groups. All of the specimens are very carefully archived in the UK CEH laboratories, and we're beginning to do some work with the Natural History Museum on DNA analysis approaches to making use of those specimens and to try and give us yet more information about the pollinator populations and indeed about what plants they've been feeding on. And there's some interesting work going on to look at the DNA side of that. So the volunteers who do these surveys for us, we provide all the equipment, we set up the square access, we provide all the maps, and we provide an initial meeting with one of the PoMS team to get you set up. But from that point onwards, there are 4 visits during the year where we ask our volunteers to take the traps out, set them up in these 5 locations across the square, leave the traps out for 6 hours, collect them back at the end of the day, collect up all the insect samples, and do a few other bits and pieces, including some fit counts in the middle part of the day. So it is quite an intensive bit of work, and we've been very fortunate to have some volunteers who've stuck with the project across all 4 years now. We have some volunteers who've been doing it ever since day one, and we do get some fantastic results from this. There is time to relax in the middle part. So they're busy at the beginning of the day putting the traps out. You're busy at the end of the day collecting them in, but there's a bit of downtime in the middle where you can go for a walk or have a sleep or find a tea shop or go back home if you live close enough to do that. And we do, over half the squares are currently adopted and have volunteers running them, but there are, as you can see on the red squares on here, are all the squares where we are still currently seeking volunteers to take these on. And some of these squares are in quite remote places where just not many people live close to them. But there's some others that are in much more populated areas, and we've we would love to get more people involved in this. It would really help us get the full set of data that we need to analyse the survey. So if any of you online, or if you know of any organisations or individuals that are near to these squares who might be interested, then our plea is to really get in touch with us, and we can let you know more about the squares. We can't promise that all of them could be seen as a holiday destination, but there are some very attractive and interesting habitats in these squares that you get a chance to go out and wander around in. So if you're at all interested in that, please do get directly in touch with us and we'll send you all the information that's needed. And finally, I just wanted to quickly say a little bit about some of the other links that the Pollinator Monitoring Scheme is making. The Pollinator Monitoring Scheme itself is a large partnership with all these organisations and funding partners from the government bodies. So there is lots of collaboration between CEH and Bumblebee Conservation Trust and BTO and Butterfly Conservation and all the others that you can see on the screen there. We have had a number of research papers that have made use of various aspects of the pollinator monitoring scheme already, and now that we have our first 4 years of data being analysed, there will obviously be more research to come out of that and more publications to come. But the methodology itself is also being used for a variety of things, and I'm very grateful for the BioLinks project, who are making use of the fit counts as part of some of the courses that they're doing around bees and other pollinators. And the fit count methodology has been picked up quite widely. Another example, last year, some of you may have seen Jimmy Doherty's Big Bee Rescue on Channel 4 last year, where they got a big community project going in Peterborough, doing lots of fantastic conservation and habitat work for pollinators and using the fit counts to do some monitoring of what was going on there. A brand new project that has its launch today is the cross-pollination project, which is particularly focusing at getting schools involved in pollinator conservation and monitoring, and they have also adopted the fit count methodology to promote as part of a wonderful package of resources that they've put together. So if you're not already aware of this project, please do have a look at their website, and particularly if you have links into schools, but there are ways for anybody to get involved in this new project that's just starting up. And PoMS is increasingly being picked up internationally as well. The fit counts are now being used in a number of countries around the world. The photo in the top right there is fresh off the press from today. There are people in Cyprus who've had a big fit count workshop today, getting people out there to get involved with this. There are proposals for a Europe-wide pollinator monitoring scheme that the PoMS partnership is very much involved with, and a similar methodology is likely to be adopted there. We're beginning to have discussions with people in the United States about how the US might set up its own monitoring scheme, and they're also looking at the PoMS methodology for that. So it is something that is sort of becoming part of a sort of global approach to how we try and keep track of what's happening with our pollinators. I haven't got time to say anything more about the Fit Count app, other than to let you know that it's nearly here. We're really excited about it. It's the sort of test versions are looking really good, and it should be out by the end of this month. We'll be making a big song and dance about it when it's ready, and please do give it a try. I think you'll hopefully find that it makes it really easy to just go straight out and do your count and collect all the data and send it straight off without having to worry about paper forms and writing things down and going to websites to fit it all in. So watch this space and we'll let you know about this one. And it has been, with the global use of PoMS and the fit count methodology, this app will be available in multiple languages over time. It'll have Welsh, a Welsh version will be built into it from the start, but it's also going to become available in other languages as time goes on and get used for some of those international projects as well. And that brings me to the end of it. I'm sorry I've gone over a bit of time. I hope we can still stop for a few questions, but please do take part in the fit counts if you possibly can, wherever you like, whenever you like, within the weather conditions. That would be fantastic to get more people doing that. If you're interested in 1 kilometre squares, get in touch. traditional species recording still going on, so we're not saying stop doing your bee recording and your hoverfly recording and the recording scheme stuff. That's a very important part of the picture as well. So lots to get involved with, lots that you can do. Thank you to all the partners and who have wonderful volunteers, and to the landowners who let us go onto their land. And that's it. Let's stop for a few questions if we've still got time, Kieran, and thank you for listening this afternoon.
Yeah, we'll take one audience question, and then we'll get Ada to the best question from the chat. So I'm going to give you a couple of seconds to figure that out, Ada. I'm just going to ask a quick question about the FitCount app, because I saw one in the chat that I think would be good to know, because a lot of people are obviously used to using iRecord for it. So Nicola was asking, will the data still go into iRecord? And will you still be able to put information into PoMS through the iRecord system as an alternative to the app, or is it moving fully over?
Okay, that alongside the new app, and fairly shortly after the new app, we're also going to have a new PoMS website. And when that website is launched, which should be in mid-May, at that point, we will stop using the iRecord form. However, all these things, iRecord, the app and the new website, are all using the same database that sits behind iRecord. So your data is still, whichever way you do it, it's all going to end up in the same place. And when the new website is available, you'll be able to add your counts to the website or to the app, but they'll all feed through, and you'll be able to go to the website and you'll be able to see the map of where all the counts are being done, and all of them will be there in one place, regardless of whether they come from the app or from the website.
Yeah. So my understanding is it's not species records anyway. So iRecord was essentially a temporary fix while you got the bespoke digital infrastructure that you needed. So
yeah, well, as I say, it's still using the same infrastructure behind the scenes, but yeah, it'll be a PoMS specific place that you go to make it happen.
PoMS has got a proper home now, which is great. Right, Jan, would you like to ask? I have no idea what Jan's question is going to be, Martin.
Okay.
So be ready for this one.
Thank you. It's a very quick practical question. You very specifically said count the insects that land on the flowers. But my garden's absolutely full of bee flies at the moment. Now, they don't land on the flowers, but they are clearly visiting the flowers. And also in the evenings, there might be hawk moths and things that don't land as well. So do we count those because they're visiting the flowers, or only the ones that literally land on the flowers?
The rule of thumb is, do they touch the flower?
Oh, okay, right.
With any part of their body. That's so when I say land, I should have said if a bee fly touches a flower with its proboscis, that's a landing as far as I'm concerned.
Fine. Okay.
The hawk moth thing is interesting because we don't generally assume that people are doing fit counts after dark, and I think actually if the temperature's warm enough, there's probably nothing in our rules that say you can't do a fit count after dark. But in practice, that's not something that we've generally sort of looked to do. So it's a daytime thing. But of course, you do get things like hummingbird hawk moths flying by day, and the same thing applies there. If it's got its proboscis out and it's touched the flower, then you count it.
Lovely. Thank you very much.
The other thing you've reminded me about that I should have said is that if you do a count and you don't see any insects at all, please still record it. Please send it in as a 0 count, because we do need those 0 counts as part of the data. I know people are often disappointed if they spend 10 minutes looking at a flower and they don't see any insect land on it at all. And I can understand the disappointment, but if you do that, please do still send the details in for the records. We do need that information.
Thanks, Matt. Yeah, it's a monitoring scheme, so we need to be monitoring zero pollinators as well as when it's abundant. Right, Ada, have we got a burning question from the chat?
Yeah, there was a question from Stephen near the beginning, sort of asking about the flowers that can be in the quadrat and whether they all need to be the same species.
Okay, good question. So what you're counting is all from one type of flower. So if you've picked dandelion as your target flower, then you don't want to count anything that's not a dandelion. So you want to define your square, your 50 centimeter square, and then focus on the dandelions. But there could be lots of other flowers in your square. That's absolutely fine. But the ones you use for counting are only the species that you've chosen. Does that make sense?
That sounds brilliant, Martin. Thank you.
I think it's fair to say as well, for those that haven't had a chance to look at it yet, when you go to the PoMS website, the guidance that you can download is pretty comprehensive, and it does go through it all in quite a lot of detail. So it's well worth having a read through them. And if you're somebody that struggles with sitting down and reading and you want something more visual, there's videos as well available on YouTube that you can watch that help explain all that as well. So yeah, there's lots of resources to go through those rules with you. Right, well, I think on that note, I'm going to stop the recording.